Okay, great. So first of all, I just want to thank everyone. I want to thank Jay for putting together such an incredible gathering. I am truly humbled to see so many amazing individuals taking time out of their very busy day. Both the doctors coming here, you guys have some patience to save, so we should hopefully make it brief. And all of you MIT alumni, I mean, it's amazing that you're able to sort of take time off during the day. And then Jay, thank you so much for putting together such a wonderful gathering, such an extraordinary group of people, and for really welcoming us with open arms. So what I'm going to talk about today is AI for genomic medicine, how to understand and reverse human disease circuitry. But our original plan was to sort of step back a little bit. So I added, the path of all of humanity in the cosmos. So, you know, as a side note. So I can't speak to this group without starting with this slide. Unbeknownst to the fact that we were actually going to be gathering, a few days ago, I was asked to give a presentation on how to blend education and research. And I couldn't think of a better place to start. I could think of no other place to start than the motto of MIT. So MIT was founded in 1861, a relatively young university. And when all the other universities had mottos that said veritas, lux et veritas, in lumine tuo videmus lumen, which basically means light, enlightenment, sitting around thinking, learning. MIT, tiny little school at the time, bunch of weird people thinking about science in a completely different way, basically said, no. Mens, the mind, when we say dementia, mens is the mind in Latin. Mens is all nice and good, and you can read your books. But without the manus, the hand, what good is education? So MIT broke with the traditional view of enlightenment and said, no, we're going to do something about it. We're actually going to learn and put the knowledge to work. So this motto of MIT, of mind and hand, of learning and doing, of theory and practice, of education and research, this mindset of MIT has been transformative, not just for MIT, but for the whole world. The educational impact of this mentality of a practical education, not just learn, but actually do something with it, is extraordinary. MIT has been ranked the #1 university in the world for many years. As long as these ratings have been around, MIT has been #1. Not yeah, go ahead.
So I'm trying to interrupt you. This is important.
Good.
Number one.
And we're not just #1, we're #1 in computer science, in data science, artificial intelligence, electrical engineering, mathematics, physics, astronomy, statistics, chemical engineering, civil, linguistics, materials, mechanical, aeronautics. Who's from one of these departments? Raise your hands. Awesome. Who's not? Come on, what are you doing? Come on. Bring it to #1. So, and also #2 in biological sciences, chemistry, economics, accounting, finance, architecture. Who's from those departments? Raise 2 hands. Awesome. So the mission of MIT is to constantly innovate in education, not just rest on our laurels, learn about theory, but actually change the way that we do education. And in fact, MIT created, in addition to the 5 schools, a college. I mean, what the heck? In 2020, MIT decided that 5 schools was not enough, and we're going to create a 6th entity, and not just another school, but a college that cuts across all other schools, so that computing permeates across every aspect of the MIT culture. MIT also had an extraordinary impact economically. I would call this the Manus part. So MIT research has been at the forefront of AI, engineering, genomics, climate, cancer, dozens of fields. It has fueled scientific breakthroughs across the centuries. The reason why, the United States survived is because of radar, core memory, understanding the expanding universe, detecting gravitational wave. All of that has been done at MIT. And Kendall Square is reputed to be the square mile that is the most innovative in all of planet Earth. We don't know of any other planets, but so far we're number one in the cosmos. MIT alumni launched 30,000 companies. Who here has started a company? Raise your hands. Awesome. So you guys are part of the 30,000 companies. Together, those 30,000 companies have created 4.6 million jobs, bringing in an annual revenue of 1.9 trillion. This is bigger than Brazil, all of Australia. This is just MIT alumni, the 10th largest economy in the world. And the examples are here, it's extraordinary, the impact that MIT has had economically. MIT has also been impactful in changing the policy, changing the way people think about research. So as World War II was ending and US economy was shifting away from the war, Vannevar Bush, an MIT grad, a student who entered in 1916, joined the MIT faculty in 1919, became Dean of Engineering and Vice President of MIT, wrote a little letter called Science, the Endless Frontier. This was very important. The phrasing is very important because the war was ending and everybody thought, great, what are we going to do now with all of this investment in science engineering that got us to win the war? What we did was increase it, not decrease it, because science is endless. Wars come and go, wars end. Science is endless. So the endless frontier, the concept of constantly pushing science further. And it's thanks to the vision of this fellow MIT alum, and I'm truly humbled by the number of extraordinary fellow MIT alums. Thanks to this fellow MIT alum, the US changed its vision for research and development centered around the university system. The concept that there's a basic research going on that combines industry and academia and hospitals and medical doctors and every aspect of society with a university as the backbone of a high quality, basic scientific research where education is key to both advance research and produce the next generation of scientists. So that led to the National Science Foundation that was started because of that. Ultimately, the NIH and the Department of Energy and all of these other funding agencies. And it didn't just change the US, it changed the world. Because just like MIT became a model for the US became a model for the world. And we now see in China and in so many other countries in the world, research and education coming together. The Bayh-Dole Act in 1980, allowed the universities to retain the intellectual property rights, the patent rights to the inventions. It's not like, oh, the US government funds the research and therefore we get to keep the inventions. No. We fund the research for free. We fund your research and it's your invention. You can then turn around and commercialize it. So I've started 2 companies at MIT and so have many, countless alumni, 30,000 companies thanks to that. But I don't want to talk about just recent evolution. I want to talk about the much larger picture of evolution. If you look at the world around us, the invention of initially RNA through an RNA world, ultimately proteins and DNA, from the cooling down of the solar system to the creation of life on Earth, it's a very short period, a few 100 million years in the 3.5 billion years of the existence of life on Earth. This is extraordinary. That allowed the concept of self, the concept of metabolism, being able to translate things that are non-self into self, being able to enable organization of the cell, of the circuitry, of communication, multicellularity, cells that instead of just each fending for themselves, coming together and saying, oh, we're an organism now. And here in China, this is not surprising. In the US, it's very surprising. The concept of collaboration, being part of the same society, being all together as one nation is, very foreign to the Western world. but this concept of working together as partners, and eventually the development of body plans, the development of photosynthesis, the development of senses, the integration of all of those senses. You can basically think of chemotaxis for bacteria as basically being able to sense in which direction is the food and turning to that food, eventually giving rise to better perception of the world. The detection of light only came because it was evolutionarily advantageous to be able to better hunt for food or avoid prey. And this ultimately gave rise to the integration of all of these different senses and the integration of knowledge, which basically gave rise to a central nervous system. And we are here today because of those extraordinary inventions. But humans almost defy these laws of evolution. If you look at humans, many extraordinarily successful humans are like, yeah, I don't want a family. I'm enjoying my career. This is wonderful. So this is very weird, right? Like the concept of you can be at the epitome of intellect in the human population and your chance of reproducing actually goes down. It doesn't go up, which is crazy. So humans, through this invention of this new type of activity, beyond just looking for food, beyond just looking for shelter, beyond just looking for mating, the new type of activity of an intellectual pursuit. This is something very novel in evolution. And again, through language, through memory, through reasoning, the integration of all of this information, we have become a new type of species. And that's also where artificial intelligence comes in. The concept of reasoning without actually having a body, the concept of being able to think about the world without being the descendants of apes and without being the descendants of evolved organisms. And something very weird happened here with evolution. The traditional view of evolution has been one of random mutations that generate diversity and survival of the fittest. But another aspect of evolution that we don't talk much about, because we are the survivors, there's something known as survivorship bias. When people say, oh, back in the days, you know, we were biking with our rusted bicycles and falling off cliffs, and listen, here we are. The problem is that those who fell off the cliff, they're not here to say, listen, here we are. That's known as survivorship bias. And we also have survivorship bias in evolution. We're like, evolution is so great because look at us. But if you're one of those guys, evolution is not that great. So evolution is coupled with extinction, with replacement, the concept of winners and losers, and winners take all and the loser just extinct. And the cost of this selfishness is when an organism becomes too good at exploiting the resources of its environment, it actually destroys its environment. And we should be a little careful as humans because we can see disrupted food chains, entire ecosystems falling apart when one species gathers too many resources. So there's many downsides to evolution. It's been slow. It's been billions of years. It's been messy. If you look at genomes, and I look at genomes every day, and they're terrible. I mean, the wiring is just a mess. It's also brutal. There's a lot of suffering. There's loss of life with evolution. It's inefficient. Every one of these species had invented extraordinary machines that can solve extremely important biological, synthetic biology problems that are now gone by the wayside. We will never recover them. So being able to understand these machines, being able to keep the good parts and throw away the bad parts is something that evolution has not done. And it's also stochastic, it's just a random search. So what's different about humanity is that we're not just following Darwinian evolution. And that's where you should interpret the people who choose to have a career instead of having children. Because human evolution works both vertically in terms of genes, and that's where traditionally knowledge was passed on from the elderly person of the village to the next generations in the village. They shared their genes, they shared their wealth, and they shared their knowledge. Evolution of knowledge was in silos traditionally, and it was all passed vertically from parents to offspring. But what's extraordinary with civilization is the horizontal spreading of evolution. An idea gets created in China, someone in Athens will hear all about it right away. And they will invent something, and someone in Egypt will hear all about it right away. And they will invent something, and someone in the US, and in Ireland, and in Iceland, and in every aspect of the world will basically spread the knowledge horizontally. And that's where all these people who don't have biological children fit in, because these people can have intellectual children. The ideas that every one of us in this room creates can spread like genes and evolve and change and mutate and grow. And the knowledge through the printing press, through Wikipedia, through WeChat, through all of these different ways of spreading information can spread now horizontally. Wealth also through taxation, through fellowships. The concept that a very rich person who didn't have children can endow a fellowship for the randomly super talented kid from Southwestern China to basically go and attend MIT on a fellowship. That's a big deal. That's a different type of wealth transfer. And if you look at the genetics, the talented individuals travel and intermix with other like-minded people. There's assortative mating. So competition has been replaced by collaboration. Sharing has allowed broad upgrades of software. And that has allowed diverse skill sets. We don't all hunt for deer and grow potatoes anymore. No, some of us are awesome at making pottery or at writing scientific papers, and there's others who go hunting and are great at that, or who go growing the food, etc. So we can basically trade and exploit our diversity, and the result is this extraordinary acceleration. If you look at the speed with which inventions have happened, we are riding an exponential. We've been riding this exponential since fire and tools and language and writing in ancient China and ancient Greece, and the printing press and the telescope and the engine, the light bulb, the telephone, the flight, vaccines, moon, Windows, creating the web, Google and Wikipedia and Facebook and the genome and YouTube and self-driving cars, and eventually general AI and being able to hit Mars. This is an exponential, and it feels really fast the last 3 months. Who feels that AI has moved like extraordinarily fast? This is just the same exponential that we've been riding for thousands of years. It just looks steeper when you're right on it. But every exponential starts out very, very slow and eventually speeds up. And we are at the speeding up point. So humans have not just been the beneficiaries of evolution, they've reshaped evolution. For thousands of years, humans have been selectively breeding animals for their properties and plants for their properties. This is teosinte, a plant in Native America where the closest relative of corn, which is the major food source in the Americas, was in fact this completely inedible plant that was selectively bred by the ancient Maya to basically give rise to corn. Wolves are, of course, the closest relatives to dogs, from an animal that will eat you alive to an animal that can't wait for you to get home, and animals that will just like die of suffocation unless we cut their wool. But it wasn't until 1860 that humans truly understood the laws of genetics, the path of inheritance. And Gregor Mendel, in an abbey in Brno, I was very fortunate to receive the Mendel Medal for genetics. And I went and visited Brno, visited near Prague, the monastery where Gregor Mendel had carried out his experiments on peas and broken the rule of genetics, and showed that inheritance is discrete, that in fact, there's no blending. At every generation, you get or you don't get one or the other version of an allele. You can have purple or white petals for a flower, and the purple petals can go for 25 generations before a white one appears completely out of the blue. Brown eyes can go on for hundreds of generations until 2 recessive alleles come together and a blue-eyed baby is born. So this is this discrete notion of an inheritance. So first rule, discrete inheritance. Second, dominant recessive alleles. Third, independent assortment. Every trait is inherited through what we now call genes. A different gene is governing each of these traits. So you would think that the world would finally say, great, we understand genetics, it's over, let's just rewrite the textbooks. Well, the paper had 3 citations for 50 years. Only a local newspaper wrote about it. and the manuscript of Mendel remained unopened in Darwin's desk. Darwin couldn't figure out how genetics works, and Mendel had sent him the answer, but Darwin never opened until his death. And the reason is that this was very difficult to reconcile with what we saw as continuous human evolution. Humans don't just get super tall or super short, they're a continuum. And they follow this normal Gaussian distribution. Why? Because as Ronald Fisher in the early 1900s and other statisticians showed, all it takes is a small number of Mendelian loci that randomly make you a little taller or a little shorter, and you randomly inherit some number of them. And as all of us from MIT know, random flipping or +1 -1 will basically lead to a Gaussian distribution. If you draw balls that basically can randomly go right or left, you end up with a Poisson distribution. And in the limit, this becomes a Gaussian. So when Mendelian mapping of traits of these single gene alleles and single gene traits was extremely successful in the 1980s, we still had no hope of understanding complex traits like height. And the reason we found out is because there are, in fact, thousands of genetic loci that influence height. Alzheimer's, diabetes, obesity, schizophrenia, every aspect of human biology is not governed by a large hammer that goes on or off. It's instead governed by thousands of needles that minutely tilt a balance one way or the other way. And that's where my group comes in, because it is ludicrous for a biologist to start studying these tiny little needles with traditional approaches. You need a completely new way of thinking about biology. At the systems level, you need to understand the circuitry. You need to understand how these are changing. So the promise is that we have now discovered 120,000 locations in the genome where we can understand that something there causes disease. And the promise is that that might give us a path to understanding causality, to understanding the mechanism, to understanding how to find target genes, to develop new therapeutics, and to enable the dream of every doctor, precision medicine and personalized medicine. The challenge is that the vast majority of these genetic loci do not perturb the proteins. They instead change the wires, the light switches, not the light bulbs directly. And these wires can be extremely difficult to understand. And that's what my research team does. We're basically seeking to start with disease genetics to understand the mechanistic basis of these disease genetics and ultimately understand what is the impact of these genetic differences at the molecular level. How do they impact RNA? How do they impact the epigenome? How do they vary in healthy individuals and how do they vary in disease individuals? And we integrate all these data sets computationally to predict what are the driver genes, regions, and cell types, and ultimately to understand this path of causality that starts with genetics and to go and test that causality by understanding the circuit, understanding how to manipulate the circuit, and reversing disease mechanisms in human cell cultures, in mouse models, and ultimately in pharmaceutical development. So to give you one example, from a few years ago, we basically went after the strongest genetic association with obesity. And we said, what does it do? And what we found is that we can trace this region of association to a single letter change. And that letter changes a T into a C, disrupts an AT-rich motif, which can no longer be bound by this AT-rich interacting domain factor, ARID5B. When the regulator can no longer bind, it can no longer repress this massive 12,000 letter super enhancer, which then turns on IRX3 and IRX5, two genes that are sitting 1.2 million letters away and 500,000 letters away. And those we found out are master regulators of a process known as thermogenesis or heat generation that basically can burn calories inside your fat cells. Instead, when thermogenesis is shut off by IRX3 and IRX5, we store every calorie that we consume and that we do not burn during the day. We store it as lipids in white fat, and that leads to obesity. This was a beneficiary allele before World War II, and this became a detrimental allele when we started having office jobs. People were not working in the field anymore. Everybody could eat at McDonald's, and people started becoming obese. So the idea now is, wait a minute, by understanding that circuit, we can figure out how to modulate that circuit, how to modify it. And we went in and said, great, can we increase or decrease the expression of the upstream regulator or of the downstream target genes? And we saw remarkably that we can switch back and forth between lean and obese phenotypes. Can we edit a single letter change and switch between lean and obese phenotypes at the level of cells? And what's remarkable is that from a single letter change, we can restore thermogenesis in risk individuals that are unable to burn calories by flipping that one letter out of 3.2 billion letters in the human genome. Flipping a single letter can restore thermogenesis. 1 in 3 billion. I don't know who would place that bet, but we placed that bet and we won. That's precision medicine right there. 1 in 3 billion chance of succeeding. And indeed, that one letter succeeded. We can put that in mice and shut off lipogenesis and turn on thermogenesis. And these mice are constantly burning calories when they're awake and when they're asleep. You can basically see here that when you put these mice in a high-fat diet, they start out leaner, and normal mice gain weight. These mice are simply unable to gain weight. So what goes on here is that the mice don't have any white fat left. They're full of only brown fat, healthy, juicy, brown fat. So no white fat. So this was the strongest association with obesity. This increases your body mass index by 1 standard deviation. This is enormous. It's rare, but it's enormous. There's another example of the strongest genetic association with Alzheimer's disease. In ApoE4 lies the strongest genetic risk for Alzheimer's. The ApoE4 allele leads to a 10-fold higher risk for Alzheimer's, 10 times larger. And individuals who carry it are almost destined to have Alzheimer's disease. So what can we do there? We can try to figure out how it works at the molecular level. So we basically carried out single cell profiling of 400 plus individuals across ApoE3, ApoE4, and homozygous ApoE3/4 individuals. And what we found is changes in every major cell type. And in particular, we saw in oligodendrocytes, these are the cells that produce the myelin that protects neurons. The axons of neurons are coated with these fat that basically allows the signal, the electrical signal, to travel much more reliably. And it also protects the neurons from damage in Alzheimer's. And what we found is that these carriers of ApoE4, even when they did not have Alzheimer's disease, even before Alzheimer's, the non-symptomatic carriers had turned on cholesterol biosynthesis by a lot. Something was happening. These individuals did not have myelin, but the cholesterol was getting overproduced. What was happening is that the cholesterol transport was not working. So the cells kept producing cholesterol to get cholesterol, but it was getting stuck in the endoplasmic reticulum of the oligodendrocyte cells. And if you look at the neurons for those individuals, ApoE3 individuals have these beautiful myelin sheaths. ApoE4 individuals simply do not. And in collaboration with Li Huitai, the director of the Picauer Institute, we basically carried out a series of experiments that allowed us to now restore myelination and restore cognition. By understanding the circuit, we basically went into human cells and we restored cholesterol transport using cyclodextrin, and we restored myelination in human cells and in mouse cells. And most importantly, when mice had lost their cognition, when they were no longer able to remember how to get out of a box or how to solve a maze, they were able to restore cognition by simply changing and restoring cholesterol transport. In another example, we went after cancer and cancer immunotherapy. And we showed that individuals who respond to cancer immunotherapy show a difference in one regulator, and individuals that do not show a different response for that regulator. So we took that regulator and we overexpressed it, and we found that indeed the cancer recurrence dropped down to 0. We were able to help the immune cells of the body fight the cancer by overactivating globally through a regulator that targets many genes, this whole pathway. So we've been able to use this type of approach of understanding the systems level view and the circuitry across hundreds of traits and hundreds of cell types. I showed you 3 examples, but we've published paper after paper, basically dissecting, in this particular paper, 30,000 disease regions in a single paper with images that have been downloaded by thousands of individuals across the world. In another paper, we basically looked at how we can predict the causal intermediates on the way to disease. And in Alzheimer's disease, predict the methylation changes that are happening at 93 years of age of an individual simply by looking at their genetics and then predicting the methylation and correlating the predicted methylation with disease to establish causality. We've been profiling at single cell resolution, not only Alzheimer's, but also cardiac disease and obesity, exercise, cancer, addiction, aging, neurodegeneration more broadly across frontotemporal dementia, amyotrophic lateral sclerosis, Lewy body disease, vascular dementia, Huntington's disease, HIV, COVID, and CMV and HSV1 human pathogens in the brain, schizophrenia, psychosis in AD, bipolar disorder, Down syndrome, autism, and PTSD. And if every one of those, we found that the same technology works, being able to sort of understand how the genetics impacts the molecular circuitry and how that molecular circuitry is altered from person to person to understand the causality, the convergence, the path through which disease ultimately changes phenotype. And that has allowed us to now expand well beyond a few dozen individuals to 430 donors across 6 brain regions to understand how all of the cell types are affected, to paint a map of dynamic progression of Alzheimer's disease, to understand the phenotypic diversity across individuals and how that relates to genotypic diversity. With new technology, we're now going into imaging and looking at the 3D positioning of individual cells in the context of the transcriptome, and then being able to use that to pinpoint in cardiac disease. The #1 killer of the planet is not cancer, it's heart disease. And being able to cure hearts, being able to reverse that circuitry by focusing on the immune cells that pick up spare lipids inside the bloodstream. They're known as mast cells. And without the spatial transcriptomic technology, we wouldn't be able to find them. Understand how exercise and diet impacts a holistic view of the brain across multiple tissues. Being able to understand human heterogeneity in the context of disease. Many people think of Alzheimer's as a disease. It is in fact dozens of different pathways that are recurrently altered. We can start breaking up complex pathways into their parts, into the pathways that define the components of Alzheimer's. And these include neuroinflammation, these include lipid transport, cholesterol, neuronal damage, DNA repair. And through this, we can now take individuals and start painting them into subtypes and understand different subclasses of individuals. And we can now develop therapeutics specifically tuned to these subtypes through high throughput methods for altering the DNA. And AI is not only allowing us to understand the circuitry, it's allowing us to now start designing therapeutics. So humans have understood amazing things. But I'm starting to think that we've only understood a tiny little island of knowledge, and that AI will allow us to cross large oceans of new understanding. If you look at drug discovery, if you look at protein folding, Maraki, if you look at protein folding, Humans have never been able to understand protein folding. We've been running physics simulations of protein folding. And these physical simulations take hundreds of years to run. We have to buy thousands of machines to run them faster. Instead, what we're able to do now with deep learning and geometric deep learning is understand the language of protein folding. What AlphaFold was able to do is get an intuition, just like a human intuition, but a superhuman intuition for what protein folding looks like. And that has allowed us to now develop these protein language models and these chemical language models that speak the language of proteins, that speak the language of chemistry. And we as humans speak the language of humans. And we've been able to project all three together in the common embedding space so that we can reason in English language what something should look like. And the machine can translate that into protein language and chemical language and help us translate. We've seen movies of people being able to speak with animals and translate between them. We are able to speak to chemicals and speak to proteins and translate between the two. So I want to leave you with a vision for a need for working together. Disease still reigns despite these extraordinary advances. We need to dramatically transform pharma from simply being able to treat symptoms when they appear and having sometimes diagnostic odysseys where patients are misdiagnosed visit after visit after visit, being able to change that, being able to move not only towards better genomic diagnosis, but prognosis, being able to enable personalized medicine through individualized combinations of treatments. Take Alzheimer's and all of its hallmarks and understand which hallmarks does every person carry. And every new patient that comes into your clinic, be able to say, well, these are the 5 things that we need to fix for this person. And there's a different set of 5 things for another person, and being able to tune the therapeutics to each person. And this requires a coalition across every department of MIT and the world. This requires computer science and biology and chemistry and biotech. This requires hospitals and patients and pharma. This also requires Sloanies and finance, being able to develop new models for financing these types of long-term developments, being able to pull together resources to go after the hallmarks, not after the disease anymore, and being able to combine these hallmarks in so many different disorders where we can reuse them time and time again. So I believe AI can bring an end to human disease and make aging optional. We can now understand the language of DNA, the language of evolution, the language of the epigenome, the language of non-coding RNAs, the language of single-cell genomics and chemistry and biology and protein folding, and really reverse disease circuitry. We can use AI to now understand the language of chemistry and design next-generation therapeutics through graph neural networks and large language models and protein language models to basically combine this ability to repurpose drugs by understanding how they work. AI will help us understand and unleash the human brain's potential, being able to understand the brain through thousands of samples, through imaging and deep learning and neuronal tracing of the connectivity, to understand the joint role of genetics, disease, and environment in shaping the human brain and cognition, how early life enrichments can lead to late life cognition, how we've in our own work in collaboration with HI, revealed the master controller, MEF2, that integrates early life enrichment and mediates the beneficiary effects in late life. Understand superagers that are resilient to neurodegeneration. And in the future, help us become masters of our own brain through learning, understanding the basis of learning, the basis of motivation, the basis of wellness and empathy and plasticity and self-growth, and enable augmentation through both external factors of what are the right treatments, what are the right interventions, what is the right music for each person, internal factors of what are the hormones that we need to change for every individual depending on the stage of their life, understanding brain waves and targeted rewiring through both reading and writing of human cognition. So I see AI as transformative, not just for training workers, but also training the next generation of students and reskilling elderly individuals who are now going to be losing the skills that are needed for the workforce. Enable through teams to train new members of the team, to enable education, enable changes as the job market evolves. I also see AI as making each of us superhuman, making every one of us Iron Man. So every one of us can have our personalized Jarvis, our assistant that is never tired, never sleeping, always thinking, always helping us, never upset, never angry, always there. So these tools are only a few weeks old. And if you look at how fast human cognition has been changing and how fast AI cognition has been changing, you can trace the path. It took us 10,000 years for civilization to get us to today. And it took AI a few hundred days to basically match the capabilities of humans for nearly every task that can be defined. So we're in for a wild ride as these tools become our assistants and our partners through these transformations. And in my own research, I have embraced AI. We have recorded 10,000 meetings over more than 10 years. Well before COVID, we started having meetings on Zoom and recording every conversation through audio, through video. I am recording the talk now, and this will be one point in this database. And this is a visualization of the embedding space projection. This is how AI thinks about every one of these meetings. Every person, every sentence, every thought, every idea can be classified and categorized and navigated through these systems that we have built. We have taken 155,000 scientific papers that have cited our work, and we have clustered them, and we're able to navigate through them and understand what each of these different clusters mean. Being able to, through interactive interfaces, be able to click on a point and say, why don't you generate whatever would be in that empty point? So we can now start mapping all of human knowledge. Every book, every poem, every love letter that has ever been written can be projected into a new space of ideas. And we can understand which of these ideas were well populated, which of them are still sparse, and how we can navigate our exploratory ships to new spaces of new knowledge. So that's what we are building in my own team, but this is one little snippet in a transformation of humanity. So I'll stop there, acknowledge my extraordinary team, thank again all of you, and really leave a view of a better humanity, a better world where we can truly seize the opportunities of AI to enable a transformation where maybe in 100 years, we'll be looking back at where the world had arrived in 2023 and said, wow, we were really just getting started. So I really hope that the next 100 years will be our best ones yet. That AI will not destroy humanity, it will lift humanity to new levels of capabilities. And I've only spoken about one part of humanity. Maria will speak about the other part of humanity.